Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we have talked about next generation sequencing, and from this class we will now introduce the technologies themselves, and we will discuss how these technologies actually have accelerated the pace of genome sequencing and how they have provided this high throughput sequencing. Okay. So, in this class we will talk about one of the first next generation sequencing technologies, which is the 454 sequencing. So, let us start. In this class we will talk about something called reads, what these reads are. We will talk about DNA library preparation, how you actually prepare DNA library for next generation sequencing. We will talk about adapters, and then we will talk about Roche 454 sequencing method. So, these are the keywords that will come across: pyrosequencing, emulsion PCR, homopolymeric stretches. So, there are several next generation sequencing technologies that are available, and these are some of them. The first one is Roche 454 sequencing, then we have Illumina sequencing by synthesis technology, we have single molecule real time sequencing from Pacific Biosciences, we have ion torrent sequencing, and finally, you have Oxford nanopore sequencing. This Roche 454 sequencing, this is currently not available. So, if you wanted to do high throughput sequencing, you cannot use this 454 platform, you will have to use this other 4, any of these other 4 which are available today. Now, despite that we will discuss the Roche 454 sequencing, because this was the first method of next generation sequencing, and you will also find a lot of data in databases from this technology, right. So, it is good to understand how this data was generated before you can actually analyze those data sets, right. So, before we go into the technologies themselves, right. So, let us talk about what the reads are. So, these are actually output sequences that you get from sequencing platforms. Okay. So, instead of saying fragments or etc., we will use this term reads throughout this course, and they can be of different lengths depending on the sequencing platform. Again, as we discuss sequencing platforms, we will talk about read lengths etc., and here are some examples, and you can have different sequences of these reads. And these collections of reads, they are actually coming from DNA library, right. So, you have this genomic DNA from which you prepare this DNA library, and that actually gives rise to these reads. Okay. So, how do you prepare this DNA library, right. So, here is a schematic of this process, full process. So, like Sanger, we start with the genomic DNA, we generate these fragments, okay, because we cannot sequence the full genomic DNA in one go, because our genome is quite big. Now, after you have fragmented this DNA, what you do is something called adapter ligation. Okay. These adapters are shown in 2 different colors here, and also by these letters A and B. You can see these letters A and B, right. These are usually 2 different adapters that are added to 2 different ends, and there is a specific reason which we will probably realize when we talk about the methods of the sequencing. Okay. Now, in this case, right, so we have added these adapters now to these fragments, and then you have something called amplification, right. So, where you amplify some of these fragments, and this is required for some platforms, whereas in other platforms you do not require amplification. And once you have done amplification, you do sequencing using these different sequencing platforms. Again, the sequencing method will be different, right. So, up to adapter ligation, this is kind of common across all platforms, but this amplification and sequencing step is as platform specific. So, some platforms may not require even amplification. Okay. All right. So, once you have this DNA library, right, you can also prepare this library using in vitro transposition. So, this is a different method than what we just discussed. Okay. So, I will just elaborate this very briefly. So, you have genomic DNA. Instead of fragmenting this, what you do is something called in vitro transposition. So, you have heard about these enzymes, transposases, right. So, these actually carry some DNA fragment and insert them to the genomic DNA at random places. Okay. So, once you have isolated genomic DNA, you do transposition in vitro in the test tube, right, and these sequences that are inserted, these contain adapted sequences, right. So, imagine you have this transposase enzyme, right, like this. So, this is your transposase, right, it carries some DNA fragment, and this is actually your adapter sequence. So, what it does, it kind of goes and inserts these adapter sequences at random places. Okay. So, once you have these adapter sequences inserted in random places, you can amplify these fragments and you can go for NGS, right. So, you can have this library preparation step, which again could be specific to the platform. Okay. So, you can use these two methods, right. You can also do this transposes method. In some cases, this probably might be the only solution that you have. So, what is the function of these adapter sequences that we just talked about, right. So, you have seen this, right. So, why do you add these adapter sequences? So, these adapter sequences actually contain primer binding sites, right. So, you have seen in many cases we need to do amplification of the DNA fragments, right. Now, how would you amplify these DNA fragments, because we do not know any sequence of these fragments, right. For amplification, you need a primer binding site, you need polymerase, right, which will start extension from this primer sequence, right. So, to generate this primer binding site, we actually add these adapter sequences, right. So, they contain some sequences for which can be used as primer binding sites. Okay. In addition, these adapters can contain something called index sequences, right. So, these are again some base pairs which enable mixing of multiple samples, right. So, you have some sequence, right, which could be unique, right, and you can use these unique index sequences for different samples. Okay. Because you can generate huge amount of data with these next generation sequencing technologies, you can sequence, let us say 96 samples in one go. Now, how do you mix them and separate out this data, right. So, to do that, you use these index sequences and you can have one unique index sequence per sample. Okay. So, if you are mixing 96 samples, each of them will have a unique index sequence, and this process is known as sample multiplexing, okay, where we can mix multiple samples with these index sequences. And once the sequencing is done, we can then look at these index sequences and separate out the samples, right. So, this actually is known as demultiplexing. So, there are two terms, sample multiplexing and sample demultiplexing. Okay, so, let us now talk about the 454 sequencing method itself. So, this was the first NGS method that was developed. It was developed by a company called 454 Life Sciences in 2006, around that time, and in 2007 it was acquired by the company Roche, and it actually was closed down. This sequencing 454 sequencing was closed down in 2013. So, we will talk about why this was closed down so quickly, right, within 6 years this was closed. Okay, so, how does this method work, right? How do you get actually the sequence data? So, 454 sequencing relies on something called pyrosequencing. So, you may or may not have heard about this pyrosequencing. We will talk about what this method is in much more detail. So, pyrosequencing, so 454 sequencing actually took this pyrosequencing method and massively parallelized this sequencing process, okay, and again we will see how this actually happened. Okay. So, in pyrosequencing, given a fragment, you have to synthesize the complementary strand. For like single sequencing, you have to have polymerase, DNTPs, etc., and in this you have to amplify the complementary strand. Okay. And these bases are called, right, when these bases are called from these light signals that are appearing during the synthesis process. So, again in a moment you will see this, how this happens, right, and that is why the term pyro comes from. Okay. So, one of the first things we do is adapter ligation. We have mentioned, right, we have this DNA fragment, right, and we can have these adapters A and B, right, at these two ends of these fragments. Okay. Okay. So, for DNA we have these adapters ligated, and once we have these adapters, what we do in 454 sequencing is something called on bead amplification. Okay. So, these are small beads on which we already have some DNA sequences, okay, like probes, okay, and these are complementary to these adapted sequences. Okay. So, what will happen is that some of these DNA molecules will come, right, and bind. So, one adapter will be complementary, right. So, they will form this double stranded part. Okay. Now, once this is done, what you can do is you can then synthesize, right. So, you can have this extension, right, from here through DNA polymerase, right. So, you can synthesize the complementary strand here, okay, and you can have this on bead amplification. Okay. So, what is this idea behind on bead amplification? It is that you are generating multiple copies of the same molecule. Okay. So, you are generating these copies in the bead from just one or two molecules. Okay. Now, this on bead amplification that leads to generation of many DNA molecules per bead, and which will be used for sequencing. Now, this on bead amplification happens in emulsion, oil emulsion, right. So, these each of these beads is separated out from one another, right. So, you have one DNA molecule, right, and one bead, and there is amplification of that molecule within that bead, right. So, only that type of molecule will be amplified within the emulsion. Okay. So, you can now generate these different fragments with as many beads as you want, right. You can have billions of beads, and each of them will contain a unique fragment, right. So, how do you ensure there is this clonal amplification? As I mentioned, right, we start with one molecule, right, and we amplify that molecule on the bead. Okay. So, you have one DNA molecule per bead and per droplet or this emulsion, right, and this is optimized through trial and error, but sometimes this happens that you cannot get one molecule, you might get in some cases 2, 3 or more, and in that case there would be some sort of signal mixing during the synthesis process, we will see in a moment, and that signal has to be filtered, right. So, we cannot use that data where you see this signal mixing. Okay. So, the sequencing happens in picotiter plates. So, these are plates like this. Okay. So, here you have these wells, okay, and there are 100,000 wells, right. So, more and each well is designed in such a way that you have one bead per well, okay, and these bead contain all these DNA molecules, right, which are clonal copies. Okay. So, copies generated from just one molecule, right. So, you can have these beads across these 100,000 wells, okay, and this is where the sequencing will happen. Okay. Now, you see how this whole process is parallelized, right. So, how do you actually make it high throughput, right. You are sequencing 100,000 molecules at once, right, instead of just one or two. Okay. So, as I said, hundreds of thousands of wells in the plate, you can have more, one bead per well, and parallel sequencing happens in these wells. Okay. So, what actually happens, what are the reactions that happen inside these wells. Okay. So, you have this bead with this molecule, okay, and as this is after the emulsion PCR, right. So, you have generated multiple copies of the same molecule, and we have these adapters here also, right. So, you can design primers that will bind here during the sequencing process and synthesize the complementary strand. Okay. So, for that we need DNA polymerase and DNTPs. So, what kind of DNTPs? These are normal DNTPs, there is no modification, just they will come and bind based on the complementary base here, right. So, which base is present in this other strand, based on that they will be incorporated in this growing strand here. Okay. So, this process, so there is a release of pyrophosphate after addition of each DNTP, right. So, any DNTP, DATP, DTTP or DCTP or GGTP. So, any type of DNTP will release a pyrophosphate if there is an addition or incorporation of that base in the synthesizing strand, in the growing strand. Okay. So, this pyrophosphate in presence of APS, right, and the enzyme ATP sulfurylase leads to formation of ATP. Okay. So, again this APS and ATP sulfurylase, these are present in these wells. Okay. They are supplied with the media or the reagent there. Okay. In addition, you have luciferase enzyme in there in these wells, and in presence of ATP and luciferin, this generates oxyluciferin and light. Okay. So, you can imagine this reaction now, this you can visualize this. So, you have addition of one base in the growing strand, right, the complementary strand that is being synthesized by DNA polymerase. There is an addition of A, immediately there is some sort of chain reaction, right. There is a pyrophosphate release that leads to formation of ATP, that is converted to oxyluciferin and light by the luciferase enzyme. This is a very fast process, but there is a light signal which could be detected by an optical detector. Okay. Now, this is the principle, right, and the detection happens by the light signal that is emitted because of these all these reactions that are happening inside these wells. Okay. And you have a detector that detects this light. Okay. So, step by step let us look at this. So, one of the questions that you might have is how do you actually identify which DNTP is being added, whether it is A, G, T or C, right. How do you identify that, or how does the detector actually identify that? Okay. So, what we do is we actually add these DNTPs one by one. Okay. We do not add all 4 together, we start with one, then wash away that one, then we add the next one, wash away that one, then add the next one, so on. Okay. So, this happens one after another. Okay. So, in each cycle we provide only one type of DNTP, right. So, these are called cycles. So, you have we are adding one type of DNTP, let us say we provide A, we see if there is any signal, and then we wash away all these A's, and then we add G, let us say we again wait for signal, right, if there is any signal or not, and then wash away G, then we continue this process with C, T, etc., and then we start again with G, right. So, this is a continuous process that happens for many cycles. Okay. So, let us imagine, let us look at this, right, how this will look like. So, how the base call actually happens. Okay. So, this is your cycle number, right. So, we have cycle 1, 2, 3, 4, etc., and this is your signal intensity. Okay. Y axis is signal, and we are following this cycle, let us say T, G, A, C, right. So, we first provide T and we see whether there is any signal. Yes, there is some signal. So, the base is T, right, we have identified the first base. Next we give G, right, we wash away T, we give G. Is there any signal? No, there is no signal. Okay. So, there is no G base here after T. Next base, next is A, is there any signal? You see, yeah, there is quite a bit of signal, and this signal probably comes from 2 A's. Okay. So, you can now imagine if you have addition of one base, you get one unit of light. Okay. Let us say if you have addition of 2 bases simultaneously one after another, right, you have 2 complementary bases that will give you, let us say, 2 bases added. So, what will happen? You will get 2 pyrophosphate, you will get everything happening in double, right. So, this should give you twice the signal intensity, right. So, twice the intensity of light that you will get if you had just one base. Okay. So, that is what we see, right. You can now calibrate the detector saying, okay, if you see this much signal, this means you have addition of 2. If you see even higher signal, there is addition of 3 bases and so on. Okay. So, let us continue the cycle, right. Next one, let us say we see a signal here, okay, and we say, okay, there is C. Okay, and then we go back, right, we go back to T again and we see whether there is signal. No. So, no T. For G, yes, let us say there is signal. So, G and so on. So, we continue this process, right. You can run this for 100 cycles, 150 cycles, or as many cycles as you want, or before you start losing the accuracy. Okay. So, this is the principle how this sequencing method works. Okay. So, you now understand that there is this chain reaction that happens because of release of pyrophosphate during addition of base, that actually translates into light, for which we can then measure this light intensity or the signal intensity, and through which we can determine the base that is being added. Okay. Now, here are the sequences that actually were present. that were available when 454 sequencing was done. So, these are again slightly different chemistries, slightly different processes, right, and slightly improved ones. Also the differences in throughput, right, how many sequences you can get in one experiment, right. So, there was this difference because sometimes you need less number of you have less number of samples, in some cases you have very high number of samples. So, depending on your need you would use these platforms. Okay. So, these are the names of them and you could use them if you wanted to do 454 sequencing. What are the advantages of 454? So, this was the first method as I mentioned, right. So, it had tremendous popularity, right. So, there were many research work that has been done using this technology. The read length was about 500 to 600 base pairs and up to 1 kb in some cases, and this was longer than competition at that time. So, let us say on average it was around 500 to 600 base pairs. It had reasonably high accuracy, so about 99% accuracy. So, that is quite good, right. So, you had only 1% error in the data, but it also had some drawbacks. Okay. So, we will talk about the drawbacks. So, it was actually expensive compared to the competition that actually came later on. Okay. So, this competition came from other technologies that we will discuss later. And why this was expensive is because it was utilizing many enzymes and many substrates for the sequencing reaction. Okay. So, as you have seen, we have DNA polymerase, but in addition we have ATP sulfurylase, we have luciferase, then we have different substrates like APS, luciferin, DNTPs, right. All these reagents, they are quite costly, and on top of that, in every cycle of reaction we are washing away these enzymes, right, and reagents. Okay. So, which means we need a lot more than that is actually required if we did this process continuously, right. So, we are losing a lot of these molecules, enzymes. So, that made this process very expensive compared to the competition that we will talk about a bit later. Okay. And you had these washing steps as I just mentioned. So, that means we utilized a lot of these molecules. There was a bigger problem with 454, that is the signal intensity was not linearly proportional when the number of bases was greater than 6 to 8 in homopolymeric stretches. Right. So, there are regions in the genome where you see a single base repeated multiple times. Okay. So, this is quite common across human genome as well as other genomes. Right. So, one base like for example, A is repeated let us say 10 times, 20 times, right. So, you have these variations across these genomes. In some places it is maybe 5 times, in some places 8 times, in some places 12 times, some places 20 times, so on, right. These regions are called homopolymeric stretches. Okay. Now, what happens in 454, as you have seen, the signal intensity goes up, right. So, if you have addition of just one base, you have some signal intensity. If you have two bases, that the signal intensity should be double, 3 bases, the signal intensity should be triple, so on, right. So, it should go up linearly. What we have seen, what the researchers had seen is that this signal was not proportional after certain length. So, after you have reached these 6 or 8 bases, beyond that the signal was not proportional to the number of bases anymore. So, which means you could not determine the exact length of the homopolymeric stretch. Okay. So, whether it is actually 10 or 11 or 12 or 14, that there used to be confusion. Okay. So, which means this method gave a lot of sequencing error around these homopolymeric stretches, especially insertions and deletions. So, we will talk about these different types of variants that we can determine. So, what it means is that we got more error in the data, sequencing data in these regions. Okay. So, here is a kind of a schematic of the whole thing, right. As you can see, okay. So, x axis is number of bases in homopolymeric stretches as I just explained, and then you have y axis, you have the signal intensity, and in the initial part up to 6 8 is linearly proportional, right. The signal intensity linearly proportional to the number of bases in the homopolymeric stretch, but then after that it slowly starts to saturate, right. So, as you go along, right, as you go beyond these 12 bases, what will you see is that this sequencer will not be able to resolve this number of bases very accurately, right. So, whether it is 14, 15 or actually 18, right, these will not be resolved. So, you will start seeing a lot more sequencing error in these regions. Okay, so, these are the references that you have utilized. To summarize, so 454 sequencing massively scaled up the pyrosequencing reaction. We have, as we have just discussed, right. This scale up process actually involved these on bead amplifications. So, we start with DNA fragments and then we do something called emulsion PCR in this oil emulsion. So, what you have is something like micro reactors, okay, and each micro reactor contains one bead and one DNA molecule coming from the genomic DNA, and this genomic DNA is then amplified because there are certain complementary sequences to the adapters already present on these beads. So, this leads to amplification, massive amplification, which we call emulsion PCR within these emulsions. Okay. So, at the end what we get is something like a bead with multiple copies of the same DNA fragment. Okay. And we have millions of such beads that we have prepared in parallel from the genome, from a genome fragment. Okay. Now, once we have these beads, what we do is we put them in picotiter plates, right. So, there are multiple wells in 100,000, you can have more, and then each well will hold one bead. Okay. So, once this bead goes in, this will actually lead to the sequencing reaction. Okay. Now, of course, in this process there will be some wells where you do not have any bead and some wells maybe you end up with 2 beads or more. Again, this can be optimized through trial and error process. Once we have the beads in the wells, we can now start the sequencing reaction. Okay. And the sequencing reaction, as we have just discussed, depends on pyrosequencing principle. Okay. So, pyrosequencing has been used before for DNA sequencing, but it was never scaled up at such a level. Okay. So, what happens in pyrosequencing is that you have sequencing by synthesis, which means you are synthesizing the complementary strand of the DNA molecule that is present on the bead, and using DNA polymerase, DNTPs, etc., you start synthesizing the complementary strand. Now, once there is an addition of a base, right, whether it is A, T, G or C, there is a release of pyrophosphate, which then through a series of reactions is converted to a light signal. Okay. So, it does not depend on the base itself, you end up with a light signal whichever the base is, and this light signal can be detected, and this indicates there is an addition of a base to the complementary strand that is growing, and this light signal then acts as the detection, right, across these wells. Okay. So, if you can visualize in your mind, across the picotiter plate the detector is seeing these signals of light, right, as the sequencing reaction is going on, it is seeing these signals of light across these 100,000 wells in the picotiter plate. Okay. And from that it is doing the base calls. Okay. And we have talked about how these base calls actually are dependent on the number of base additions. This should scale linearly, the signal should scale linearly with the number of bases that are added. Okay. This method is highly accurate, as has been observed, it is about 99% with comparable read length to Sanger sequencing. So, Sanger sequencing gave about 900 base pair to 1 kb read length, this method gave us about 500 to 600 base pair at most. And then what we have seen is that, of course, this is not perfect, there are some drawbacks. So, one of the drawbacks is it's very expensive because it is utilizing so many enzymes and so many reagents, and it could not actually compete with the newer technologies that were much more affordable compared to this 454. So, this is the reason why 454 was closed down in 2013, it could not compete with the other methods. But we have also seen that these sequencing errors are biased. So, we have seen that there are homopolymeric stretches where this method does not perform that well and you get higher error rate. And we have just mentioned this is more expensive compared to competition, and that is why this method was stopped, but nevertheless it had a huge impact. It actually allowed researchers to do a lot of fantastic work, and even today you will see a lot of data in databases around, and in case you analyze this data, you need to keep these drawbacks in mind, especially for example, if you are doing this looking at homopolymeric stretches, you need to be very careful with the analysis. That is it for this class. Thank you.